Okay, so today I'm going to be talking about the life cycle of fungi. I'm primarily just going to be talking about the asexual stage and the sexual stage. The asexual stage is fairly simple. They primarily go through mitosis, and that's about it. Fungi can reproduce asexually through a few different ways. They can go through fragmentation, producing spores or budding. And fragmentation is essentially when a piece of hypha or mycelium breaks off and will just grow into a new mycelium. Depending on the type of fungus, there would be one nuclei in the hypha, which would be divided by septa. Those are kind of those cross walls. In other fungi, there might be multiple nuclei. There might not be septa. For the sake of the video and what I drew out, I just drew out one nuclei divided by septa. When we're thinking of mitosis, especially in asexual reproduction, when we're thinking of mitosis is what? It's when one cell divides into two identical cells. So the nuclei, the haploid nuclei will undergo mitosis and will divide into two identical nuclei. Those will get packaged up into spores and those will go out and germinate into the wild. And that's about all there is to it with asexual reproduction. The sexual stage of the fungal life cycle is a little bit more complicated. First step would be plasmogamy. I misspelled that on my drawing, but plasmogamy is essentially when two compatible fungal cells fuse their cytoplasm. So the cytoplasm of two compatible fungal cells will fuse. This will happen with different mating types. So with fungus, there isn't female or male, there's positive or negative or plus or minus. So if a plus or a positive comes in contact with a negative strain, those would be considered compatible. In plasmogamy, they would fuse, producing a cell with two nuclei. So it wouldn't be a diploid cell yet, it would just be two haploid nuclei. That would be called a dikaryotic cell. So di meaning two, karyotic meaning nucleus. So you can kind of see in the drawing in the hypha, you have your just two nucleuses there, but they're not fused yet, they haven't combined. In karyogamy, the two nuclei are actually going to combine, creating a diploid cell. Fungi don't usually spend a lot of time as diploid cells. They usually just go under meiosis pretty fast. So plasmogamy is when the cytoplasm forms. There's two nuclei. Karyogamy is when those two nuclei actually form, creating that diploid cell. Next step would be meiosis. This is when the diploid cell is going to divide into two haploid cells, or I'm sorry, two haploid nuclei, and it's going to divide into another two haploid nuclei. So in total, you're going to get 4 haploid nuclei. This all happens in the same cell. Like I said, the diploid cell will divide into two haploid nuclei, and then those two haploid nuclei will divide into another two haploid nuclei. So in total, you're getting 4 nuclei. Those 4 nuclei are going to get packaged into spores, so like zygospores, basidospores, and then those are going to get germinated out in the wild. They're going to get dispersed by wind, water, etc. They're going to germinate and then they're going to restart the cycle essentially. So that's all there really is to it. By far the sexual stage is a little bit more complicated than the asexual stage. The biggest thing for the asexual stage you got to remember is that it undergoes mitosis. The sexual stage undergoes meiosis along with plasmogamy and karyogamy.